Okay, so for the last little bit, we're going to turn our attention to a real-world data set where, the goal, where we actually, or where the people doing the measurements actually did this workflow of trying to find a full discovery data set inside of DIA data. So no longer targeted to a specific pathway or complex or some small cellular function, but to actually target as much as a DDA run would target. And that's, that I think rightfully they realized that DIA we were getting a lot of blowback from people saying, why would I ever use, something that's not going to detect as much as DDA? And so this was a pretty path-breaking publication for me, certainly. And that was the first time that I had seen this full DDA library applied to DIA. It's the Brooder paper that's been mentioned a few times, MCP 2015. And then there was also, Mike mentioned the Celebsec paper that happened the same year. And then we followed that up the next year with the Navarro paper, where we included a bunch of tools and showed that they could all get the same results. But so we're looking at Brooder. And yeah, I want to just quickly remind, so this was I would say the first time that I really went, oh, now I get what they're talking about. This was the first time where I really got this, oh, we can do DIA for quantitative samples and do pooled or fractionated or something, even on the same instrument, to build a library and then go back and look for those things in the library. It's the whole thing that Mike was talking about of we can pinpoint something in some other samples, very precisely with higher resolution data, whether it's using DIA or DDA or whatever, but we can learn information, which are the spectra, the relative ion abundance in fragmentation, under fragmentation of the molecules, and their retention time. We can learn that information and just apply it on a really broad scale and actually get more out of the samples than we can get by doing the single, the way that the field was doing things is we would just do these single shot things where we would do DDA and we'd get as much as we could get out of DDA, and then we would quantify based on MS1. And I think that this was really to me, the sort of expanding the scope. And obviously they felt like they really wanted to compare against DDA. I think this story is really compelling, but I also think that they oversold it a little bit in the paper. And so this is the figure that Rudy showed, and it shows wow, we were able to detect all this stuff in DIA so repetitively, and then DDA not that repetitively, and then we've got this the number of things that could get detected in every sample is going down. And but I do want to point out that in this paper we also have this statement. So it says on average, we found 28,600 peptides in HRM, and an average of 17,500 peptides were identified per measurement in shotgun proteomics. But you also have to realize that this is the critical sentence. It says no feature alignment was used. And so nobody does that. And that's kind of a little unfair, because nobody does does this MS1 quantification without feature alignment. MaxQuant certainly never does. That you would have to, try hard to turn it off. And this is a DDA experiment inside Skyline that we looked at, or I was talking about before I was showing you slides of, and you can see that basically, so that map with all the holes, here's the case where this particular peptide was detected in 4 out of 9 runs, right? You can see a little ID shows you whether or not it was detected. And then if I turn on peptide ID times aligned, and I allow for alignment, I guess it's not that easy to see unless I do maybe this. So then you see these light lines that are the aligned ID times into other runs, and that's how, and we have beautiful peaks. So we can still quantify these things, even though but it's true that the mass spectrometer, for whatever reason, it must have been busy with something else. It never sampled this peak, or it was a chimeric spectrum that never got identified, or whatever. Um, so but clearly we can still quantify this. So it's not quite as bad as like, oh, we have to, in order to quantify this data, we have to use imputation. We're not really using imputation because these aren't real missing values to the statistics, because we do alignment and we actually find these. So this was, I think, a little bit overstated. I still think it's a great story, and yes, you can get better quantification out of DIA, you can get lower limits of quantification, all these things, but this figure to me is a little bit overstating the point. And so, but the Brooder data set is definitely extremely interesting to look at for us. And just to remind everybody, so where we are now is what I call the prior knowledge workflow. So we can do DI acquisition. We can do this at any time. We don't need to do it. We don't need to collect the spectral library before we do DI acquisition. We can do DI acquisition of our samples, knowing that we're going to quantify using this method. And then we can create a spectral library outside, use IRT to normalize, and then now we can do this product iron targeting that we've been doing, and chromatogram extraction. And then we can now use, well, we used mProfit peak picking, but we can use it on a very much larger scale and do quantitative analysis. We're going to actually, this is in Skyline, we have a spectral library and an IRT library, and you've done that, I think, in the tutorial. There's also this thing called an assay library, which gets shared around and gets used by these other tools. So all 4 of these tools were shown to do very well, and produce very similar results. And we're going to work in Brooder. We're going to work with an assay library. That's what was published with the paper. So we're going to just import that directly into Skyline, and then also we could use a chromatogram library from Panorama. But the principle is very much the same, that you do these separate runs to get the coordinates that you're then going to go query in your DIA. So let's just get started with, so for this experiment, turns out there's a bug in 4.1 in handling of assay libraries. So I'm going to have everybody run Skyline from the flash drive that I gave you. Here's a little secret about Skyline. You don't need to install it. You can, if you have a full Skyline installation in a directory, you can just go run it from there. So let's go to it's called side-by-side installation. So as long as you have a full set of Skyline executables in the right place, you can just go run it from there. So we'll go to USB. Hopefully everybody has USB installers, and then we're going to go to Skyline Daily 64-bit. And now here's like a setup exe and everything, but we can just keep going, drill into application files, into Skyline Daily, right? 4.1.18827, and then we just have to find Skyline exe and double click on it, and now we are running Skyline Daily. And we're actually running a specific version of Skyline Daily. So even if you have Skyline Daily already installed on your machine, you're using the version that I created explicitly for this All right, and so I even what I did, I actually put this whole path into a command prompt so that I can bring it up just by executing that command, which you can do as well. So anyway, so okay, so we're ready. We're gonna go to a blank document. And so here is Skyline with a blank document. If you drop the settings menu, you should see default. Okay. And so that's what I was saying, like, oh, we should put that at the top of our tutorials is like start with settings default because we had so many people having like, oh, this one setting is set and I missed it. And so if everybody starts from, and that's why I created settings default probably at some point after that tutorial was originally written, and just so that everybody could start on the same page. All right, so the next thing I'm going to tell you to do is just turn on integrate all. Right, we're not the only time you would want integrate all off is if you're actually doing a refinement experiment, which is not what we're doing, and otherwise it's going to mess us up. And we're working on fixing that because I have to teach this a lot. Turn on integrate all. Okay, so then we go to peptide settings, and we are, well, let me actually give, before we get too deep into this, let me give you an overview of what we're trying to bring in. Uh, so on USB, there is this DIA pub, and so we're going to get into both the Brooder publication and the SlevSec publication, both from 2015. In Brooder, you'll see that there's a library folder, and in the library folder, you can see both these XLS, these Excel spreadsheets are kind of the original documents. One is the IRT reference sheet that I downloaded, and the other one is an assay library as an Excel spreadsheet that actually was in the supplementary data for the MCP uh, paper that I just showed you. And then, so then I took a specific, this is a multi-page, this Excel spreadsheet is a multi-page thing. I think both of them are actually. I turned them into CSV files, and I'll just quickly open one. So this is the assay library, what is known as an assay library. It's got a modified sequence, a stripped sequence, the precursor charge, protein ID, protein description, et cetera, and then it's got it's got the IRT value here and the relative fragment intensity. So that's sort of the spectral library, but it's a very stripped down spectral library. You just built a spectral library out of search results, and you got actual measured spectra in it. And at this point, you sort of have, I think of these more as decorated transition lists because I come from an SR, I started this with SRM, but it has these just the main additions to just a normal transition list that you would see in SRM are this relative fragment abundance and the IRT value. So we're going to import all of this into Skyline. If you open this, close it down because as we learned, and don't save, as we learned with somebody here, if you have a file open in Excel, nobody else can open it, and we're going to want Skyline to open this thing. So if we go back up here, then we have just one raw file. There's actually 24 raw files in the experiment. And I have that all on a flash drive. If you want to get really wild and crazy with this stuff and you want all the raw data, you can come to me. It's quite a bit more than I've given you. But we have one raw data file. And then we have scripts. And I'm going to show you how I use these scripts later. But they run Skyline command line. And they're extremely useful. I use some variant of these particular scripts quite a lot. I showed you that plot where I had varied retention time and I'd varied the extraction width, and you could see the number of detections going up, and that was all done sort of running Skyline command line overnight. And I'm going to show you how to do that. And then oh, this is hands-on. This is what we're about to build. So I'm just gonna delete that one. That was just me practicing. Um, and then we have, this is the fully imported data and a template, and but we're gonna build our own. Oh, and then this Brooder data settings just gives you an overview of what we're about to do, these settings that we're about to do. And then there's an MSstats, this is an exported report from a fully processed document that you can process in MSstats. And hopefully we'll get there. So if we go back to our Skyline Daily, we're going to keep trips in and we're going to allow for 2 miscleavages. And because we're importing an assay library, some of these settings don't matter as much. Like Skyline will just override the miscleavages. If your assay library contains peptides with miscleavages, Skyline will just go, well, that's what you want. But I think it's good to just sort of document what we're doing, even though these don't really restrict Skyline when it's importing an assay library. In the prediction, we're not going to set up a retention time predictor quite yet, but we'll go ahead and say that, hey, if retention times were present, we're going to use a 10-minute window, even though that's not really going to matter. We're gonna set the length from 6 to 55, which is what I've determined actually shows up in this assay library. Again, if you import an assay library with 55 amino acid long peptides, Skyline is not going to use this restriction to say, no, you can't import that. It'll just override it. And we're also going to get rid of this exclude N-terminal amino acids. So 6, 55, and 0 there. We don't have a library. No library should be checked by default. I don't even have any. You might, well, I guess everybody just started a new Skyline Daily, so you probably don't have anything here either. And then we use a fair number of modifications. So go ahead and, when you're on the modifications tab, click Edit List, and then click Add. And the first one you can just type oxy, and then drop the list. Whoa, I didn't. Oh man. Oxy. And then, wow, it's not picking it. I don't know what's going on. Oh, oxy with an I. I should have just stuck to OX. Okay, so OX, and then when you drop the list, we want oxidation M, methionine, and then just okay that. And then we're gonna add, you can type ACE and drop the list, and it will choose acetyl, and we want acetyl N-terminus, not the lysine one. And then we okay that. And then now, this is where we're talking about losses. They used water loss and ammonia loss, which I think in some cases inflated the number of ions they were able to find and use. But you can just type AM and drop the list, and then you do ammonia loss KNQR, which are the only those are the only amino acids that allow ammonia loss. So click OK, and then we're finally going to add just WAT, and now we get water loss on D, E, S, and T. And those are the only amino acids that allow water loss. So you should now have a list that looks like this. Anybody need more time? Raise your hand if you need more time. Oh, we need more time. You must have changed something. Cancel and start again. Because those defaults, if you leave them alone, they'll go right in like the way they did here. Okay, so once you've got all those, click OK, and then go ahead and check them all. So those are the modifications we're going to allow when we import the data. Everybody got that? And click OK. We're done. We don't need to go to quantification. And then transition settings. Again, as I mentioned this morning, for those of you who are here, I usually, and when I'm doing full scan, I'll just start with full scan and set up my full scan settings. We're not going to do MS1 extraction. We're just going to try to keep things small and fast. If we do MS1 extraction, it doubles the number of transitions we extract, and this is a big enough method already. But I still highly recommend using MS1 extraction, just not in this case because it's a pretty big set already. So let's go ahead and choose DIA. And I will also point out that the whole Navarro paper used no MS1 extraction at all. So it's definitely, it works and it produces comparable results. So here we want to use 15 ppm. So we're going to stay with centroided extraction. So what I also found in Navarro was that extraction from centroided data works quite well. 10 is a pretty good default. It works on a lot of Q-exactive data, but apparently we need a little bit wider on this data. And now we're going to go define a new isolation scheme. So the ones that we have here aren't really right for this one. We mostly have swath, DIA. And what we want is exactly the one that was used with this data, right? And so we're going to use a pre-specified isolation scheme, and I don't even know what it is. So I'm going to import it from one of my files. So Skyline, if you point Skyline at a DIA file, Skyline will figure out what was used in it. So we're going to go to, click import, and then get to your USB DIA pub Brooder raw1. So you want to get to that raw1 folder where you see this one raw data file. This is sample 1 replicate 1. So this is the first raw data file in the set of 24. And I just put it, the only reason I put it there was so that you could do this. So is everybody at the raw1 folder? Yeah, it's in DIA pub, Brooder, and then raw1. And then you'll be there. Got it? Okay. So what? Oh, raw1. BD1, it's sample 1 RO1. Okay. So click OK, click open. And Skyline will then take a second or two to think about it as it looks at this raw data file. And then it extracts the isolation method, and this is off a Q-exactive, and you can see that it has one MZ margin. So that's different from the traditional swath method had 0.5 MZ margins. And then we can have Skyline graph that for us. So click graph, and you can see that it's one of these variable window methods. So I'm going to expand it, and you can see these overlaps, these little margins. We could turn them off, and then you see that, okay, as far as Skyline's now we're showing extraction windows. So if we click the margins, we show what's going to be measured by the mass spectrometer. And if we turn that off, we show what Skyline's going to be extracting from. And you can see that there's no overlap in what Skyline extracts from. There's just overlap in what the mass spectrometer measures. And that can be derived in the same way as the original swath paper. So go ahead and close that. This is a nicely formed isolation list ranging from 400 to 1220, and the initial smallest windows are 30 MZ. So these are wider windows than the original. They're wider windows than the original swath paper, and they're definitely wider windows than are getting measured today on the TOF instruments, the 6600 the current SciX instruments. But this was 2015. Okay, so we're going to just call this 400 to 1200, and this is 19 variable windows. So that's the name of our isolation scheme. Click OK. And then we're going to be telling Skyline to use predicted retention time, even though we don't have any way of predicting retention time yet. So we're gonna tell it do its extraction 5 minutes on either side of some predicted retention time that was gonna come from our assay library, because our assay library has IRTs. So then we're gonna import the assay library and we're gonna get a retention time predictor. Okay, so that's all we need in that tab. In the instrument tab, we're gonna change the range to 1800, so 50 to 18. Oh, actually, we're gonna go 300 to 1800. This is actually a trick I learned. I guess that. What do you guys use, Ben? You use 350 or 250 or what? For fragment ions? What's your minimum fragment ion? 350. So they usually use 350, and that's so we've been. I guess I haven't necessarily shown you this, but in the MacLean lab, we'll frequently start by saying we only want ion 3, which means Y3, and we'll exclude Y1 and Y2. Another way to get that kind of exclusion is to go with a minimum MZ, and then you'll just get no small ions, which can also be, if you're allowing a lot of charge states, that can be pretty important, because a quadruply charged Y3 can be really small. And in this experiment, they did allow quite a bit of they allowed very high charged fragment ions. So that's all we need to do, 300 to 1800, and this is just another way of saying I don't want the stuff in the grass that there's gonna be a lot there and more likely to have interferences. On the library tab, we'll say that we want 6 product ions and a minimum of 6 product ions. So this is, what we know works very well. It's from the original swath paper, and what the Aerosolid lab uses a fair amount, I understand. And I guess we'll leave everything else the way it is. And then on this page, filter page, again, this is, Skyline will import whatever the assay library has, but I'd like my settings to sort of reflect what it has. So I'm going to say 1, 2, 3, and comma. So you do 1, 2, 3, 4, how high are they going to go, 5, 6, 7. They actually have charge 7 precursors. And these, this is all, the assay library comes from MaxQuant, so they must have allowed MaxQuant to find charge 7, to match charge 7 precursors to the spectra. And then you also see fragment ions in this data set that go 1, 2, 3, 4, 5, 6, up to 6, charge 6 fragment ions, and that's why it is pretty useful to have a low MZ cutoff, because a charge 6 fragment ion could definitely be down around, small ones could be down around in the tens of MZ. And then we're going to allow B, Y, and precursor ions. And we're going to say ion 1 through last ion, because we're not going to have any extra filtering other than the 300 MZ. So Skyline, we would typically, or the MacLean lab would typically not accept anything under ion 3, and we're also typically only allowing charge 1, maybe 2 fragment ions. And then we're going to turn off this special thing. We'll go ahead and leave this use DI precursor window for exclusion. Do people understand what that is now? Did you use it before? Anybody not understand it? Okay, I'll explain it. Uh, it's basically saying that, you know, if my isolation window is 400 to 425, don't pick a fragment ion in there because there's going to be a lot of precursor stuff flowing in there anyway, and there's a higher probability of uh, interference from unfragmented precursors that came through. So we decide not to use that. So because and this is something you can only do when you have a predefined isolation list, right? But again, this is mostly documentation because we're using the assay library, and if the assay library picks an ion in the middle of there, we're going to go ahead and use it anyway. We won't exclude it based on this. So if we were importing a FASTA file the way you did, it would be more important. So if we built our own library inside Skyline and then imported a FASTA file, these settings mean a lot more. But okay, so that's it. We don't need anything in prediction. And so if everybody's set up like this, is everybody set up like this? Okay, let's click OK. Ion charges must contain separated. Oh, I got an extra comma in here. So now if I click OK, library count must be, somehow I got back up to 3. I don't quite understand that. Did anybody else get that? Library, yeah, okay. So there's some kind of bug when you switch a value. I think it's probably when I'm switching these values, it somehow sets the default back to the default, which was 3. So I think now we ought to be able to go done. Everybody got those settings? Okay, so let's go ahead and import the assay library. So file, import assay library, and it's going to tell us, no, it isn't. Okay. Uh, so we'll pick, so I came up, so if you didn't come up in, you want to be in Brooder 2015 library. And you want this MCP M114 CSV file. Anybody need time to find that? So did you come up pretty close to there? Okay. So click open, and this will take a moment. So anybody who's behind can try to, and then okay. So now it tells us that the document needs to be saved, and we're going to save it up in the Brooder 2015 directory. You can see there's a brooder.sky. We'll call ours hands-on brooder.sky and save. And then Skyline should get to work, and it starts importing the assay library. So does anybody have any questions at this point? Because it takes a little while to import the assay library. Do people have it in importing, or people are, yeah, it's done? All right. Mike, I need a new machine. Okay. Yeah, I hope you got the right file. Okay, so did you get to this 329 errors? Wow, that was fast. Okay, so what are these 329 errors? Well, so first of all, what? 6,000. So you missed some steps. But so yeah, we can. So everybody should get 329. If you didn't, then you missed some steps. And so there must be some setting. So who has anybody gotten to the here 329? Okay, wow. And has anybody who didn't get 329? Oh, wow, quite a few people. And okay, so what did you get? What? 20,000. Yeah, so there's a lot of rows in here, and every row can potentially cause an error. Uh, so one nice thing that I put in recently, because it's sometimes hard to, these error messages are pretty descriptive. It says, hey, this precursor MZ does not match the closest possible value. And then it gives me the peptide, and then it says, check my modifications, et cetera, et cetera. But so you can actually just, and I used to go, oh, let me go look at line, it's line 13,695. So I'd go open up a text editor, but I added this checkbox at the bottom so you can see what did the line look like. And actually in this case, you can see right away, okay, Skyline just cannot handle an N-terminal acetylation and an oxidation. It can't handle two modifications on the same amino acid. So that's the problem. So every peptide that looks like this, and there are a few, not going to make it through Skyline. We're not going to worry about it. So let's go down to this next one. Here we see, and this one says, wow, the precursor MZ 400-242 of the peptide is outside the range covered by the method. Now, does everybody agree with that? Or do you think that the range covered by the method actually covers that? Yeah, didn't it start at 400? But wait a second, it had a one MZ margin. So if we went back and looked at the measurement windows, yeah, we measured 400. We measured 400, but we don't want to extract out of it. So this is an error in the assay library. We're extracting out of that margin range, or we're expecting that we can, and so we could go back and move the measurement range and pretend that we measured from 399, although we got that range from the raw data file, so we didn't actually measure 399. But we could go back and pretend we did, and then we would get all of these and we could go extract out of that margin. So it turns out that if you do that, that accounts for 250 of these are within this 400 range. So I think we don't have a huge amount of time. We don't have any time barely. So we're not going to go back and do that, but we could, and then we would get down to only 88 errors, and we would find that the majority of those, so these are still, if you keep scrolling down, you can see lots of 400s, and then the majority of things that aren't the 400 error are this doubly modified methionines on the N-terminus. So you can pretty quickly get to an idea of what the errors are, as long as you don't have 20,000 of them. Yeah, so what is that? Product ions. Yeah, I haven't, if you had a whole lot of them, I would worry about them. In this case, I'm not going to worry about them. In the past, I found that it's like, oh, that they were even more lenient on losses than I thought. So there's actually Hannes Roast, one of his papers. He was using an older version of Spectrast, which apparently allowed like for 4 to 6 different kinds of losses, and then it allowed up to 3 losses on a fragment. And I just thought that was, unlikely, and it's not the way we build the assays anymore. And so my guess is that, yeah, there's some reasonable explanation for why these fragment ions can't be found. So yeah, product MZ value 93732, and you can sleuth them out and figure out what they are. It just takes work. So this is just evidence of the tools not quite matching up. And usually, like, I was amazed when Ben gave me the pan-human library, I imported it and there were like no errors. And so I think we're getting better and better at having the tools agree on what are valid ions and what are the best ions to measure. And so this is 2015 assay. We're just going to keep going. We're going to drop 329. 250 of them are within that 400 range, and 88 of them are explained by this double modification on the N-terminal methionine, et cetera. So just so go ahead and click OK. Sorry, those people who had 20,000, I can come look at that in a moment, but what? You got 329? Oh, and what was it? Ah, yeah, okay. That would explain it. Okay, especially the if you don't select the water losses, it turns out there's a lot of loss ions in this thing. Okay, so now we get the Skyline knows this is an assay library, but it says like I don't see your IRT standards, so can you tell me where they are? And one way would be that the assay library itself might contain a protein. So sometimes you'll see right at the top of this list IRT, and then you just choose that. But in this case, it didn't. So since it's coming from Biognosis, we use the Biognosis standard values for IRTs, and we just get those from a separate list by clicking that and then clicking Browse. And then right in that same library folder, you can see this Biognosis IRT CSV, which I created from the product sheet. That you should have gotten to here right as soon as you closed it. This should have been the next thing. Did you cancel or? Yeah, okay. So if you don't have any targets at this point, you better re-import. Okay, so then go past the errors, and when you get past the errors, go ahead and do separate transition list. Everybody got that? Okay, so then, and then we choose this Biognosis IRT CSV. That should then be in here, and we can just click OK. And this again will take a second or two as Skyline does what it does. The longest part is it actually builds a whole spectral library out of all those relative ion abundances. And this is building a little SQLite file. And then the really frustrating part is that you're like, oh, it's almost done, and then it says, oh, I'm finishing up, and it just finishing up. It's like, well, how long are you going to take to do that? I don't know. It takes a little bit, but then we will actually have the vast majority of the Brooder assay in there. But we're going to see that remember how we chose 6, we wanted 6 transitions per peptide. And Skyline's not going to enforce that for us when we import an assay library. So we're going to end up with however many were in all of those files that we just imported once it finishes up. Any questions? Then mine. Technical limitation. Yeah, I mean, you can. Yeah, I was sort of looking at one is I would be doing this on a desktop. It's just going to go lots faster on a desktop. Even this computer I was looking at has like a 2.5 gigahertz processor, and I'm used to being closer to like 3.7 or 3.8. And I was just I always buy a computer that's like ever since I graduated from college, which is an extra long time ago that I won't go into. I always get a $2,000 computer. Like that's what Microsoft was buying me in the 90s, and it's still about what I aim for is to spend about $2,000 on a computer every 2 or 3 years. And I was looking at what you can get for $2,000 on the way here, and it's this Dell XPS tower, and it's got like 64 gigabytes of RAM. It's got a hexa core, so 6 cores instead of 4, and they're all running at 4.6 gigahertz. So this is what what I intend to go convince my boss I need. Uh, and what I think you should convince your boss you need too. And then these things will just scream. But we're dealing with yeah. So anyway, so we're dealing with this with all of our laptops, which are a little slow, but we're doing our best. Okay, so we've got everything. You can see the IRT peptides here. You know, they have the little clock faces, which is what we would want. They have 3 transitions each, which is how Biognosis defines them. And then if you look at this next protein, you can see, well, that's more than 6 transitions. And you look at the peptides, and then you can select the peptides, and you can see here's the spectrum that, so Skyline has created this pseudo spectrum. It's not a measured spectrum. You can't see any noise. It's just the relative ion abundances, but you can get a sense for the relative ion abundances and the rankings, and what transitions they correlate to. And you can see that Skyline's matched all this up, and everything's looking pretty good. But let's go ahead and enforce the 6 transition limit. Actually, do we really have to crash on out of here right now, or what's my hard limit? They don't have to be there. I'm the one that's going to be late, right? I can just catch a lift over. Yeah, I'll keep going with these guys, right? I mean, we told you it was going to be till 6 o'clock, right? Yeah. So yeah, I don't want to cheat them. All right. Just because you couldn't get a late dinner reservation. No, anyway. So I'll get myself over there. Okay. So we want to actually enforce this 6 transition, but before we do, let's go to the document grid and create a report that will show us how many transitions all these things have. Okay, so the first thing we're going to do is, so that have people been using the document grid? Did you get to use it in the tutorial a little bit? Okay, so did you customize a view at all? Anybody customize? Okay, so we're going to create a custom view, and a great starting point is you could either start from some other custom view, or there are these 5 5 just basic views. Like you definitely went to the replicates one. We don't have any replicates. We're going to start from precursors, because the question we want to answer is how many transitions does every precursor have, right? So we're going to start from precursors, and then we're going to click that same views menu and customize that view. And we got to give this a name. We're going to call it transition counts. So this is a new thing, and we're going to get rid of a bunch of these columns. I'm going to get rid of results summary. I like precursor charge. I don't have any, it's unlabeled, so I don't need the isotope label type. I don't need the neutral mass. I don't need the collision energy. I like the modified sequence. And I don't need the precursor note, library name, or library type. So I've got those 5. Just delete columns until you have these 5 columns. What? So at the top is a little views thing. That's where you chose your precursors, and then you drop it again and do customize view. Which one did you delete? Which one did you delete? Because you can always put it back. What? Precursor charge. So just expand peptides, precursors, and then it'll be right there, right at the top. Oh, there's undo. Yeah, we put undo in here. There's undo everywhere in this product. I forgot about that. Yeah, just undo if you deleted something you didn't like. Okay, the other nice thing about here is people are like, oh, how do I find that column? Hey, there's a little binoculars. There's a way to find things. So we can just type count, and that will select the transition count as a column. So I'm going to go ahead and close the find window and check transition count, and then it puts it above the modified sequence, but I want it at the far end, so I can use the arrows to change the order. So everybody should be able to get to having precursors, peptides. Oh, I want one more column. I actually want, so I'm going to select precursors, and then I want proteins too, so I can see what protein the precursor is. So I should have proteins, precursors, peptides, precursor charge, precursor MZ, modified sequence, and transition count. And as you can see, you should explore this. There's tons of values and some really great reports you can make once you understand this. Okay, does everybody have the transition counts report? I can see some people still scratching their heads, so. What? Yeah, so we have these first three proteins, peptides, and precursors all checked, and then some other precursor values, etc. So click OK. Everybody ready? I see some people still looking at their screens. All right, well, you can look at the columns once I click OK because they're still here. Protein, precursor, peptide, precursor charge, precursor MZ, and modified sequence. And really, as long as you get precursor and transition count, then you have some information. You can see that we can see that all the IRT peptides have only 3 transitions, which is not a problem. They won't even get included in our M-profit model. And then you can see 9s and lots of 9s. And so let's go ahead and sort by transition count, which we can do by clicking on the transition count column and then filter. Oh, not filter, sorry. Cancel out a filter if you did filter. Transition count sort ascending. And now you can see that, well, the IRTs are the only ones that have 3, but then there's one with 4, and there's several with 5, and some with 6. And actually, if we'd allowed more of those 400 MZ precursors, there would be more with 3. Those 400, we saved ourselves some really small peptides by not allowing those 400 peptides. But you can see, so most of them have 6 already. There's some 5s and 4s. So let's go ahead and get rid of those by going to Edit, Refine, Advanced. And before we do this, it's usually you know, an okay idea to have a look at the numbers down in the status bar here to tell you how many proteins, how many peptides, how many precursors, and how many transitions. So, and then we're going to set that we want a minimum peptides per protein 1, so that if by some reason, for some reason, this removes some peptides, we'll get rid of our proteins. And we're going to say we want a minimum transitions per precursor of 6, and then we're going to auto pick all of our transitions. So that's all you want to do. 1, 6, and transitions. And the nice thing about this form is all the values that you don't fill in, it just ignores. So if you brought up this form and didn't fill anything out and clicked OK, it would have no effect. Yes. If there's more than one, oh, you mean this? Yeah. Yeah. Right. No way. All right. Well, just hit enter, I guess. That's really unfortunate. Yeah, some of these we definitely have had some problems with some of these higher res monitors scaling and things disappear, but sorry about that. Okay, so is everybody ready to click OK or press enter? Yeah, so if you don't have buttons, enter is OK and escape is cancel. We hopefully have that working in most places. But go ahead and take a screenshot of that and send it to us, and we'll try to get it fixed. And this takes a second because we got a lot of stuff. It's actually removing quite a bit. And then finally, my machine is now done. I wish there was some progress with that rather than just freezing up like that. But now you can see, thankfully, this actually took work and was added fairly recently. We don't get rid of the IRTs, so we won't remove any of your standards with refinement. So you can refine to your heart's content and never have to worry that it will get rid of any of your IRTs or any QC standards or any standards of any kind will be excluded from your refinements. But you can see that everything else now is 6. right? We got 6 transitions for everything else. And that's what we wanted. And so I'm going to close the document grid. You can see that I have, okay, this is the moment of truth. I have 175 transitions and 175 175,227 transitions. Yeah, all right. Some other people got the same thing. It's different from my notes. If you actually look at my notes, you'll see a different number, and the different number in my notes is because I went back and changed I went back and changed 400 to 399, so I actually got those 400 MZ peptides, but most of them appear to have gotten removed because there's not really that many more transitions. But you can see we didn't lose any proteins. We lost some peptides, a few precursors, and definitely a lot of transitions. Okay, so that is it. Oh, the last thing we're going to do before we would import data is to go. So our importing data. Before we import data, we also want to go edit, refine. So that's the assay, right? Is this 175,000 transitions, and then that's we're measuring 29,000 peptides, right? So that's a pretty good, pretty comprehensive set there. And then we're going to go to add decoy peptides. So you did this before. You want to do this right before, you don't want to do this too early. This happened to somebody in here was if you generate decoys and then you modify your targets, go generate decoys again because your decoys will kind of stick to your old targets. So you want to do this when you're like, yep, assay looks great. I want to go import some data and actually run mprofit and everything. So click OK. All right, so now we have the full assay. We can go ahead and click Save to save it. And you can see that we have 350,000 transitions, right? So 350,000 transitions. That means every 3 runs, that's 1,000,000 chromatograms. And we have 8. So we're going to have 8 million chromatograms when we're done here. So that's pretty good size, right? But yeah, we've worked a lot to make Skyline pretty fast at doing that, although, it still takes time. So I haven't given you any raw data. So go ahead and save this file. That's your end of working with this file because you don't have the raw data to import. You have that one raw data file. You could import that on your own time. Or you can come to me and get the flash drive and get the full data set and try to process it on your laptop if you want. But we're going to turn our attention to, so let's go ahead and start that. So go ahead and file, so save it and then open. And you got the hands-on Brooder, but we're going to open this other Brooder, which you can see is quite a bit bigger. Our hands-on Brooder is 56 megabytes, and the other Brooder is already over a gig, and that's because I've imported data into it. So, and then there's chromatograms also in a SCID file. So let's get that started. So just click open on that thing, and that will take a little bit of time to import because it's got so much data in it. But while it's importing, I'm going to direct your attention to, has everybody got that? Okay, so it's imported, it's opening this big file. And I'm going to direct your attention to my remote desktop. And I'm going to show you, can I make it slightly bigger? Which slightly bigger? Oh yeah, this is not important. I'm starting my remote desktop, and hopefully it will be big enough to see it. But it takes, I don't know what, something happened so that it now takes a longer time to connect to my remote desktop. So we're going to take a look at some faster computers. Yeah. I'm on EduRoam. Yeah, it happens at home, and I don't know. There's some sort of like log me in used to be able to connect to my computer faster. I don't know why. Um, but so we'll just wait for that, and yeah, there it is. I had hoped that I would get to this quickly enough that it wouldn't time out on me. But so here is my remote computer. You can see that it's got 16 gigabytes of RAM. It's got an i7 running at 3.6 gigahertz. Boy, do I wish I had that 4.6 gigahertz. That's quad. Because then I would have 12 of these. So I only have 4 cores, but they're hyper-threaded, so you see 8. So that's this computer. And then if I tunnel another remote in to we have this big server, and I had to get onto that one too. And so here you can see that we got 192 gigabytes of RAM, and this is, who can tell me how many processors that is? Well, it's 48. Well, it's actually running a little bit, the gigahertz is running a little bit slower. So that is kind of surprising that if you don't get enough parallelism going on this computer, it can actually be slower than my desktop. So it's really important that we do things in massively parallel on this computer, but we can do things massively parallel. And so what I'm going to show you is these are the scripts that you have now with, they're each one of our pubs data folders has a scripts folder. And there's this multi-analysis.bat where you have to set up some, can people see that? Probably not. You have to set up some system specific values. You have to tell it where your root analysis directory is. You can see that for me, it's D Brendan X 2018 NEU Brooder of 2015. And then now I'm pointing to my Skyline command exe, Skyline CMD exe. It's just the same type of, installers, Skyline daily application, etc. that you have. So you could set this up totally on your system. And then I have to point to R. And that's just going to program files R and figuring out what version of R you have and an R script. And so this is because this whole process, this script runs through taking a template like we just created, importing data into it, building an mProfit model, selecting peaks, exporting a report, and then running an R script on that report. And so you can use this to generate the MSstats report, or you could use it to generate anything. So I use this a whole lot. And then there's some other things that are not that there are a couple other sets that end up being pretty okay for everything. And then finally we do a call to this batch script, which is Skyline analysis.bat, and we give it where the raw data is, a place in that raw data directory to put this trial, and then the .sky file. And so but I could have 20 of these, go to bed, and wake up in the morning, and if my computer is fast enough to do 20, I'm all done with 20 different analyses, and I can see how they compare. I can use an R script to generate, Excel spreadsheets, and so I can do lots and lots of processing without having to sit there and hunt and poke on Skyline to get it to do it. So this is the Skyline analysis.bat. This is where the parameters come in, 1, 2, 3, and then this defines how many processes I want to run. So this is saying, and I'm saying 12. I'm saying I got a big computer. I want to run a lot of parallel processes. So we talked about, somebody was asking what's the difference between many and several, and it's usually like 2 and 4. Well, this is 12, and we're going to see how that goes. So actually, let me get it started. So while we'll get it going, and all I have to do is I'm in So here I'm in the Brooder 2015 folder, and I just call scripts multi-analysis.bat and hit enter, and it says, hey, I'm running this trial assay lib 6 ppm, etc. So if you have a little bit of programming knowledge, you can definitely modify this batch file to do all kinds of things. If we switch back to here, it'll tell us, hey, the import log changed. And then we can see that already Skyline has, while I was talking, it opened the file, and then it saved it to a new location, and then it started a new analysis, and it told me that this report already exists, but it's resolving the conflict by overwriting, and then it got the new report that it's going to use, and then it opens the file, and then finally it just adds the results. And then you can see that we've been saying that Brooder has 24 raw data files. Here they all are, I think. Yeah, 24. So we're going to do them in 12 at a time. And if we switch over, if I now press F5 to refresh, you can see Skyline has started going. It's got 10% between all these 12, and then it's getting down to 16%. So it's running the processing. And if we switch back here, at this point, you look at it and you go, hey, you're not using all the processors. And that's true right now, but we'll see. We'll see by the time we leave here, this CPU is going to be pegged at 100%. So at this point, this early point, it's doing a lot of data reading. I think the disk is not really, I don't know. You can't see how well stressed the disk is. But mostly it's just trying to figure out where the IRT peptides are right now. So once it gets over 40%, then it's going to start really doing a lot of calculation. You're going to see that that more of the CPU gets used. So let's take one more look at this. You guys have been looking at our script, so I think you should be able to look at batch files. So what do we do here? We say, oh, if a skid file exists, we delete it. That's all, and we have various sort of running trial. And then yeah, so the first thing we do is save to a new location to allow for parallel processing. So we don't want to open our old thing. And there's actually some go-to places that will sort of jump you to the middle if you run into an error. Sometimes you want to jump past some stuff. And then, so first we save to a new location, and anytime we get an error, we go to the end. And then now we do the import, and you can see that this is a pretty long command line in Skyline. You can see that we've got a timestamp, a MemStat stamp. We import with a number of processors, directory, and import all the data in that directory. There's a naming pattern, and then we import a report, pcarriers.skyr, overwrite resolution, report name, precursor, pcarriers scored, report file is here, and then we want to export report invariant, which is the format that a lot of is easiest for our scripts to deal with. And then that should get us the report, but there's also sometimes you just want to export the report again, or you just want to run the R script again. So there are little places to jump to do that. So in most cases, I'm going to come through here and I'll jump over this report because I already did the report above. I don't need to start Skyline again and report again. And then I just run an R script. And it's called detection.r, and you can put whatever you want there. So, and then you write an R script, and that's it. So let's see how Skyline's doing on that. So it's into the 40s, 50s. It's into 68%. So it should be using a lot of my CPU. There it is, 100% of my 48 core server. So and that took quite a bit of work to get it to use that much. There was and definitely the original implementation, because Skyline was built for SRM, kind of used maybe 1 or 2 cores and really didn't stress it. But now we can really just, if you use the command line, we can totally use every ounce of your CPU. You can see that we're not using very much memory, right? We got tons of memory, so we could do much larger jobs in this. Um, no, not this one. No, this one's like tens of thousands. It's like $12,000. It's a server, and I mean, like, I actually got the idea by going to MaxQuant, and MaxQuant has like what you should buy to really run MaxQuant, and it's like something like this. It's a Dell Edge, and actually the Aerosolve Lab has one of these too, and so I just started going like, well, if you had one of these, we didn't have one of these at the time, but I was like, if you had one of these, I want to make sure that Skyline can use all of it. And I think we've done that. And yeah, you can do quite a lot of processing. If you want to do big scale DIA processing, you should definitely learn how to use these scripts and use Skyline command line. Okay, we'll come back to that. Yeah, the scripts are in the folder. So I'll show you that in a second. So I also want to get my other computer started on this same task. So here's my other computer, and I'm also again in the Brooder. In this case, it's C test Brooder. And if I look at the scripts, you can see that I localized these top 3 lines to, now I've got R3.4, and I'm in C test installers and C test Brooder 2015. So you could set this up. I mean, this is how all of the files that we're about to open up with imported data, I did them on my laptop with these scripts. So you also can. I have a full SSD hard drive that has all the data. If you have enough space on your computer, you can come get what? No, it's not on there. The raw data is much bigger. These files are like 4 gigs a piece. So, yeah, you think about it, 24 of those, you got about 100 gig. So it's about an extra 100 gig. If you've got it, I can help. I can give it to you. Yes, at this point, the server has to run Windows. We have it in our sites and in our last grant to try to get this over to. There's .NET. There's a new version of .NET that'll run on Linux. And so we think that the command line version should be able to run on Linux, but we haven't done it. We haven't done that work yet. If you know a developer who might be interested, just send them our way. Okay, so that's it. Oh, did I start this? Okay, so let me start the analysis on here. And then if I go back to here, you can see that this instance of Skyline is now saving out its file and opening that new file again, and then there it's got the 12 files, and it's going to start importing too. Okay, so we now have 2 machines doing the same analysis, and we can go back to our work. So somebody asked, do I have those scripts? Yes, so the scripts, both Brooder and Celevsec have a scripts folder. So, and inside the scripts folder, we've got the peak here is SKYR, which is the report, the Skyline report. There's also this MSstats report plus .skyr that you could set up. And then there's multi-analysis.bat and skyline.bat. And multi-analysis.bat, once you get it up, you can have a line per analysis, and you can just have it cycle and create and do a whole. I mean, if you have the disk space for that, it's got every analysis is consuming disk space. So, but anyway, so you have that, and these are very handy. I've used these same scripts a lot, just tweaking them for different uses. Oh, one more thing back here. is that you can see in the Skyline analysis.bat that I have said I want 6 processes. So instead of, I can't do 12 on this machine, I don't think, but I can maybe do 6. And if I look at, it's already off. It's not using 100% yet because it hasn't gotten to that 40% mark of the import. So this is extracting the IRTs, but it's already using 75% of my CPU. So, all right. So back to here. Let's see if we can get through this. Okay, so if we go back to Skyline, does everybody have this open? Okay, and so this is kind of these replicate plots. If you have the IRT protein selected, you can see these very distinct IRT peaks sticking up, and they're all labeled. And then you can look over here, or if I expand this, these are the IRTs, and you can see that they are detected very consistently in retention time space. Yeah, you can switch here. I just did this recently. You can switch the value to retention time, and then you get a line plot. And you can see the lines going really nice, straightly along. I'm going to switch back to value all, and then you get the bar plot again. And then if I pull out this, you realize this once again. We showed last time that there was a case where a peptide, and maybe actually it was the last eluting peptide too, in that other sample, that really dropped by 200-fold. This is sort of a 2-fold drop, or the other one dropped by like 100-fold. This is a 2-fold drop. But a lot of things drop, and this is clearly there's something went wrong with this run. We could let's go ahead and right click here and order by acquired time. Oh, and now you see this is the acquired time order. So this is the order they were acquired, and you can see there's quite a lot of variance in peak area. So you wouldn't want to use these as normalization standards, right? Because it's really unclear that anything is doing this 2-fold up and down. So logically, so the acquired time order, if we go back to document order, that would sort of tell us, I think, that sample 6 through 8 are somehow prepared differently from the other one. That's my theory. And then also this sample 2, something happened with sample 201. It just makes it a little different. And so that was like somehow in the sample preparation something happened, and it's not just because of the acquired time, it's not just like the instrument stopped collecting the late eluting peptides or the chromatography changed or something. All right, so that's just I like to look at the IRT peptides and just sort of make sure that they seem sane. But so now we do have we have our IRTs, we have our decoys at the bottom, we have everything imported, we have the 348 transitions, and we're going to go ahead and go edit, refine, reintegrate, and this is what you do. We're going to do what you just did again, add, and this is a big data set, so it's going to calculate peak scores, peak group scores. So so who can do the math? How many peak groups does it need to calculate scores for? Well, we said there was going to be, so there's 300,000 transitions, which means there's 8 million chromatograms. And then how many peaks in each chromatogram? Well, as many as 10. So this is potentially 80 million scores that it's got to put together and get ready for this training. So that's why it takes a little bit of time. But hopefully everybody can get through it. Anybody have any questions at this point? I went add. So I clicked add so that it starts calculating this, and then it puts this up. And everybody should have just like these this 9 or 10 scores. And we're using decoys and profit, and just click train model to get Skyline to keep working. So it starts training the scoring model. So this is where it's doing linear discriminant analysis. And thankfully, it used to do 30 iterations of this no matter what, but now we kind of look at it and we decide when it's converging. And it never does more than 10 because I found that really that extra 20 didn't matter. So it goes a lot quicker. It converges after 5. And then now it's got to calculate the scoring contributions. So it's got to get those little percentage values, and it does that all in parallel. So this will sort of jump to 2/3 done in a moment. Oh, I guess it did. But it goes pretty quickly because it's actually all in parallel, and they come in and fill this, and there we go. And yeah, so this is our model. You can see that shape is our best score. That's often the case. Library intensity dot product is a really good score. Retention time difference is not a very good score. And we could go back and look at that. We could use some of the regression plots like I was showing and try to figure out, well, why are the retention times not, maybe there's some curvature again or something like that. Intensity is actually never that great a score for DIA for sure. And then coelution count is actually just something that I made up. It's the original Skyline score, and somehow it ends up usually being about as good as the shape. But yeah, before we had models or any sort of complicated scoring, the coelution count was just the number of transitions that appear to be colluting, and that's a pretty good predictor. So does everybody have that? Okay, so we'll go ahead and save that as Brooder hands on and click OK. Yeah. Oh, red? Yeah. So the red just means that this is going the opposite direction of what we expect. So basically it's saying, oh, I've got a negative coefficient for that. So that means that really good signal to noise is a bad thing. So you could, yeah, I'm sorry. I don't think it matters much, especially when we're down at like point something percent. It's just like, this is not a meaningful score. I don't quite know what direction to put it in. So, I don't think it would matter at all. You could uncheck it and then retrain the model for sure if you wanted to. But it's not really a big issue here. Okay, so click OK. And then we just let Skyline reintegrate the peaks by clicking OK again. And this takes some time. It actually starts out working, doesn't know what it's doing, and then now it just goes and adjusts the peak boundaries based on the scores. Yes. I'm taking them all. Most of the tools will deal with the Q values. So Skyline is marking everything with Q values. If you choose that, take only Q values, only peaks, what it's going to do is it's going to go zero out a whole bunch of peaks. So they're going to turn into NAs. So like Skyline's going to say, yep, that's missing data because it's below a certain threshold, which is totally valid, but I prefer to just keep the values and let the statistical tools, the MSstats report includes the Q value. So it can make the same, it can do things based on the Q value. It doesn't need to have me remove the peak area. But yeah, I mean, basically it means if the Q value is less than that, then or more than 0.01, then we don't really trust the peak area. And so one option is to say, well, if you don't trust the peak area, get rid of it. But it's also informational to see like, well, what did Skyline think was the best peak, even if it was a low Q value or a high Q value. All right, so I'm almost done with adjusting peak boundaries. Anybody else done? Some people are using remote desktop. Are people using remote desktop? No, on your laptop. That's great. Okay, so while I'm waiting, I'm going to switch back to my remote computers. And you can see, well, I guess my CPU is still not 100% on my desktop. I can go back to this import log and press F5 to see an update and see where it's at. And you'll see that, it's just now getting into, it's on its second batch of 6 files, and it's just now getting into the 40% range, which means my CPU usage should be going up to 100% like it did. Okay, so then if I go look at this other computer, I think it might actually be, well, yeah, it's actually done. So if I go here, yes, and I can go to the end, and you can see that this is output from the R script. And you can see up here is the end. It's actually saving, and here's where it built the model, and it shows me this model should look very familiar, right? This is our 20, 46, 25, and our negative coefficient. So this is the same model. So we just got the same results, just running a script, and now we actually then exported a report and generated a PDF. So let's go look at the PDF. So where I look for this is in this raw folder. Here is a separate folder for this analysis, and inside the folder you can see the SKID. You can see peak areas.csv and a summary.csv, and even this peak areas no quant CSV, CVs, and then we get this PDF. And that's all that stuff is generated by the R script based on the peak areas scored CV as my report. So we haven't exported a report yet in our manual processing. Like what I'm doing on my laptop, I've gotten most of the way there, but I haven't exported the report. What I'd really like to do is export an MSstats report, but we're not going to get to that. But so let's open up the PDF. Here's the PDF. It shows me how many detections there were, and you can see it's around 27,000, or I think that's probably, and then the mean is 25.9 thousand detections per run. I don't know how that compares with the manuscript. And then you can see that we don't really have a false discovery rate problem, because as Ben showed, like it stays pretty flat. And then this is, we go out here, we see that there's 19.7 that are detected in everything, all runs. 19.7 thousand are detected in all runs. This is the number that have CVs below 20%, but that was before I started doing normalization. And so if you do normalization, that number goes up to about 17,000 CVs less than 20%. And then there are more plots in here. And this is another one that tells us we don't have a false discovery rate problem, because this is the number that are in every run, and this is the number that are just in 1 or 2 runs. And if there were a false discovery rate problem, you'd see this would spike up here. And we definitely see that. Like when there's a false discovery rate problem, this thing becomes 2 distributions, one over here and one over here. But because we were looking for things that we'd already seen in DDA, we didn't see that. And then this is looking at the CVs, and you can see the CVs changing as I require. The CVs get better and better as I require them to show up in more runs. and then actually then I do normalized CVs, and then this is 2 dimensions showing the area. So you can see that the peptides with smaller peak areas have larger CVs in general. And when we go to when we go to higher peak areas, we get better CVs. That's what that's showing. Okay, so that's all PDFs generated by R. Totally, that script is really nice. This guy's still working and will be for probably another, but this one, so that one processed everything in about 15 minutes. This will process everything in about 45 minutes. Your question? Okay. Um, so yeah, we're at 6 o'clock. Uh, so everything is processed. I'm now interested in just last thing before we leave. I we based on the usefulness of some of those graphs, we've been adding some of them to Skyline. So like you can go to peak areas and look at a CV histogram. So go to Skyline peak areas CV histogram. Mine shows up with my peak areas. Here it is. And you can see with no normalization, this is what my CVs look like. And they're this good because most of this is background, right? 24 runs, most of what we're measuring is background. So the things that are supposed to have signal are not supposed to have low CVs. But let's go ahead and try median normalization. And that makes things a little bit better. But still, like I only have 60% below. Let's also go into I can right click here and choose properties. So right click in the graph and choose properties, or there's a little properties button over here. And then I can set a Q value cutoff of 0.01. And I think that's all I need. Maybe I'll make the font size a little bit larger so you can see it better. And there we have, with a 0.01 cutoff, I get a little bit better. I could actually just, I'm going to go with maximum CV of 100 so that I'm zoomed in a little bit better on the part that I'm interested in. And then I'm allowing anything that was detected in 2 runs, and I can increase that. So you can see that I can go all the way up to 24. So these are the things that were detected in all runs. And when I normalize, then my CVs, 80% of my CVs are below 20%. So that's getting pretty good. Getting better anyway. It's not the most fantastic I've ever seen, but so you might think about like, well, okay, maybe I should I would really like to measure things where half my runs, where the thing is detected in at least half my runs, and the CVs are less than 20%, and that would cut down the number of peptides that we have to look at a lot. And we're going to get to that tomorrow when we start looking at CelebSec data. But that's it for this. The next thing we would do is file, export, report, and we would export an MSstats report. Oh, we would go to document grid. Yeah, we would have to actually annotate the replicates the way we annotated them before. And I have the stuff to do that, but we don't have time. So that's it for today. Again, I'll be here at 8 o'clock in the morning. 8 to 9 is a time where if you want to do more work on this data set, we can, or if you want to go back to the DIA that we were doing yesterday, we can. Kind of open to the people who show up. But then in terms of the main course, we're going to switch over to the CelebSec data set.